Welcome to Aurelius Explainer. Tonight, there is somewhere I would like to take you. Not up into the bright and the noisy and the endless scroll of the day, but down, slowly down, down past the warm surface water where the light still lingers, down past the place where the blue begins to deepen into something darker, down past the last faint suggestion of the sun until there is no sun at all. We are going somewhere very deep and very old and very quiet. And the strange thing, that's the thing almost nobody tells you about this journey, is that the deeper we go, the calmer it gets. Not more crowded, not more frantic, not full of teeth and terror, quieter, emptier, gentler. The deepest place in the southern half of our planet is not its most crowded. Life grows softer, not louder, the deeper you fall, and that is where we are going tonight. So, settle in. There is no rush. We have all night. Let me tell you where we are going first, so you can picture it as you drift. There is a long, narrow wound in the floor of the Pacific Ocean. It runs for more than 2,500 kilometres, beginning near New Zealand and curving up towards Samoa, a great dark seam in the crust of the world. We call it the Tonga Trench, and it is deep. It is so deep that it holds the second deepest point known anywhere on Earth. Only one place goes deeper, the Challenger Deep in the Mariana Trench, far to the north. Everywhere else on the entire planet, every other trench, every other dark valley in the seabed falls short of the place we are going tonight. In the whole of the Southern Hemisphere, there is nothing deeper. This is the bottom of the southern world. And within the Tonga Trench, there is one spot that goes deepest of all. It has a name, a soft and fitting name for a place at the edge of the known. It is called the Horizon Deep. It was named after a research vessel, the RV Horizon, from the Scripps Institution of Oceanography, which crossed this stretch of ocean back in the early 1950s and measured what lay below. Horizon, the line where the sky meets the sea, the line you can never quite reach because it keeps receding as you move toward it. A good name for a place that almost no one will ever see. The horizon. Deep lies roughly 10,800 meters beneath the waves. Surveys put it somewhere between about 10,800 and just under 10,900 meters, because measuring depth from the surface is not a simple thing. You cannot drop a tape measure into the abyss. You send sound down into the dark and you listen for it to come back, and from that long, patient echo, you work out the distance, give or take a few tens of meters. So we will say it plainly and gently, about 10,800 meters down. More than 10 kilometers of water above the deepest floor. That number is so large it slides off the mind, so let me give it to you a different way, a way you can actually hold. Think of the tallest mountain on Earth. Think of Everest with its summit at 8,849 meters, scraping the edge of the sky, so high that climbers need bottled air to breathe up there, so high that planes cruise near its height. Now imagine, if you could, lifting that entire mountain, all of it, base to peak, all that rock and ice and altitude, and lowering it gently into the horizon deep, base first, until it stood upright on the bottom of the trench. You would expect the summit to break the surface, surely. A mountain that tall planted on the seabed must poke up into the daylight. It does not. If Everest stood on the floor of the horizon deep, its summit, the very peak, the place where the air runs so thin you can barely live, would still be roughly 2 kilometers underwater. 2 whole kilometers of dark ocean above the highest point on land. The trench could swallow the tallest thing on the planet and still have room to spare. That is how deep we are going tonight. So let us begin the descent. Let us go down slowly the way you would in a small submersible, the way only a handful of human beings ever have, a journey that takes hours, not minutes, because the deep does not let you hurry. At the top, the water is bright and busy and full of life. Light pours in from above, and where there is light, there is growth. And where there is growth, there is feeding. And where there is feeding, there is movement everywhere you look. But as you sink, the light begins to change. The reds fade first, then the oranges, then the yellows, until the whole world is a deepening blue. And then, somewhere below about 1,000 meters, the blue gives out entirely. The last of the sunlight, faint and tired, simply stops. From there on down, remember, we have more than 9,000 meters still to fall. It is permanently, totally dark. Not dim, not shadowy, dark. The kind of dark that has never once known a sunrise, the kind of dark that has held still for longer than there have been eyes to see it. Below 1,000 meters, the sun has never reached, and it never will, and yet there is light down there. Just not the sun's light. In all that blackness, the only light is the light the animals make themselves. Soft, cold, living light, sparks and glows and faint blue flickers, switched on and off by the creatures that carry them. We call it bioluminescence, which is a long word for a simple, beautiful thing, light made by life. A little chemical flame inside a living body, used to lure, to startle, to signal, to find one another in a world without dawn. Picture it as you fall. The dark all around, and here and there, far off, a single soft glow drifting by, like a lantern carried through a wood at night. That is the only light in the deep. The animals are their own stars. As you descend, the water grows colder too, but not in the way you might fear. It does not turn to ice. Down at the bottom of the trench, the temperature settles somewhere between about 1 and 4 degrees above freezing. Cold, yes. Cold enough that you would not last a moment unprotected, but not frozen. The deep is held just above the freezing point of seawater, warmed a little, strangely, by the immense pressure itself and by the faint heat seeping up from inside the earth. So it is cold and dark and still, but it is a steady cold, an unchanging cold, the same cold tonight as it was 1,000 years ago and 1,000 years before that. There are no seasons down there, no storms, no day, no night, just the long, patient, even chill of the deep, holding everything in a kind of slow suspension. Now, before we go any further down, and we are still in the upper reaches really, with the deepest stretches still far below us, I want to ask you something gently. Wherever you are listening from tonight, in whatever corner of this large and quiet world, I would like to know. Some of you are listening from cities, some from small towns, some from places I have never heard of, all of you somewhere out there in the dark, drifting towards sleep. Tell me where in the world you are down in the comments whenever you wake up or whenever the mood takes you. There is no hurry. And if you are new here, if you have just arrived at Aurelius Explainer for the first time, you might think about subscribing so that the next slow journey finds you when you need it. That is all. That is the only thing I will ask of you tonight. The rest of the night is just for sinking, because that is the promise of this place and the promise of this night. Tonight we are going to go all the way down, not skim across the surface, not glance at the deep from a safe distance, but descend slowly and completely into the horizon deep and stay there long enough to truly see what lies at the bottom of the Tonga Trench. We are going to meet the animals, and there are animals, real ones, soft and pale and patient. We are going to understand how anything at all can live under so much weight, and we are going to discover the strange truth that turns the whole story upside down. that the very deepest point is not the busiest, not the most monstrous, not the most alive. It is the emptiest. Life thins out as we approach the bottom, and the reason why is one of the most elegant little secrets in all of nature. But we will come to that. We have all night to come to that. For now, keep falling, keep sinking through the dark, past the soft glows, into the cold. There comes a point on this descent where the ocean stops being merely deep and becomes something else entirely. Below 6,000 meters, the water enters a realm with its own name, a name borrowed from the oldest stories we tell. We call it the Hadal zone. Hadal, from Hades, the lord of the underworld in the old Greek myths, the dark place beneath the world where the dead were said to go. The naming is fitting. The Hadal zone is the underworld of the ocean, the deepest layer there is, and it is found in only one kind of place. It is found in the trenches, the vast flat plains of the deep seabed. The abyssal plains bottom out at around 6,000 meters. To go deeper than that, you need a trench, one of these narrow, plunging valleys where the crust of the earth is being pulled down and swallowed. Trenches are rare. They cover only a tiny fraction of the entire ocean floor, little more than a scattering of dark seams across the whole vast bottom of the sea. And yet, here is something to hold as you drift. Those rare trenches contain something extraordinary. Although they make up only a sliver of the seafloor, the Hadal trenches account for the deepest 45% of the ocean's entire range of depth. Let that settle. Nearly half of the total depth of the ocean, measured from its deepest point up toward the surface, belongs to these strange, scarce underworld valleys. They are uncommon, but they reach further down than anything else, and so they hold a kind of monopoly on the truly deep. When you want to find the bottom of the world, there is nowhere to look but a trench, and tonight we are inside one of the very deepest of them all. So picture where we are now. We have fallen past the sunlit surface, past the fading blue, past the last of the light at 1,000 meters, down through the long, dark middle waters, past 6,000 meters, into the Hadal zone, the ocean's own underworld. And still we have not reached the bottom. Still the horizon. Deep waits below us, thousands of meters further down, in cold and dark and silence. The pressure is building. The water above us is stacking up, kilometre upon kilometre, a weight beyond imagining. And down here, in the underworld, the question that has followed us all the way down finally has to be asked. Because everyone knows the old line about the deep sea. Everyone has heard it. The pressure down there is so enormous, they say, that it would crush you in an instant, crush a submarine, crush almost anything we could build. And it is true that the pressure is enormous, more than we can really feel in our bones just from a number. So you would expect, wouldn't you, just kind of that nothing could possibly survive it. You would expect the bottom of the trench to be a dead place, a sterile floor where the weight of the world has pressed out every last spark of life. And here is where the story begins to turn, because it is not a dead place. Things live down there. Soft things, pale things, patient things, things that swim and swarm and grow and feed in the crushing dark. The pressure does not kill them. They live their whole lives under a weight that would flatten us without a thought, and they do not seem to mind it at all. Which leaves us with a real puzzle, a true mystery, the question that we are going to carry with us for the rest of this long, slow night. If life can survive the pressure, if the crushing weight is not, after all, the thing that ends it, then what does set the limit? Because there is a limit. Something stops life from filling the very deepest point. Something thins it out as we approach the bottom. Something keeps the last stretch of the horizon deep almost empty. If it is not the pressure crushing the animals, then what is it? What is the boundary? What is the line that life cannot cross, down there in the dark? Hold that question. We are going to answer it tonight fully and gently, and the answer is far stranger and far more beautiful than crushing pressure. The answer is not brute force at all. The answer is, of all things, chemistry, a single, small molecule, and the quiet limit of what it can do. But we are not there yet. To understand the limit, we first have to meet the animal that pushes right up against it. We first have to meet the deepest fish on Earth. So let us keep falling. Let us go down to where the snailfish live. There is a creature down here that has earned the title of the deepest living fish reliably known to science. It belongs to a family called the Liparidae, which is a tidy, scientific name for a soft and unlikely-looking thing. We call them, in plain English, snailfish. And if you are imagining something fearsome, some great-jawed monster of the trenches, all teeth and glowing eyes and menace, let that picture go.
Let it dissolve in the dark. The Hadal snailfish is nothing like that. It is one of the gentlest-looking animals you could meet at the bottom of the world.
Picture a small fish, often only 20 to 30 centimetres long, about the length of your forearm or less. Now picture that its body is not firm and muscular and silver like a fish from a market stall, but soft and gelatinous, almost jelly-like, with a pale colour somewhere between faint pink and translucent, so that in places you might almost see through it. It has no scales. Where you would expect the tough armour of a fish's skin, there is instead bare, smooth, slightly baggy flesh, loose-fitting like a coat a size too large. Its skeleton is reduced, partly cartilage rather than hard bone, lighter and more flexible, less of it than a shallow water fish would carry. Its eyes are small and reduced, almost an afterthought, because what use are eyes in a place where the sun has never shone? In permanent darkness, vision earns its keep no longer, and so the snailfish has let it fade. But if it cannot see, how does it find its way? How does it find its food? It feels. It tastes the water. Along the side of its soft body runs a sensory system called the lateral line, a row of tiny detectors that pick up the faintest movements and pressure changes in the water around it, so that it can sense a ripple, a stir, a nearby body, all without a single ray of light. And it smells and it tastes. Chemoreception. The ability to read the chemistry of the water, to catch the faint scent of food drifting through the dark. So it moves through the blackness, not by sight, but by feel and by smell, a soft, pale shape, drifting and sensing, sensing and drifting, reading a world it will never see. Now here is the thing that should surprise you, the thing that begins to unlock the whole mystery. This soft, fragile, almost translucent little fish lives under pressure that would, by every instinct you have, seem certain to destroy it. And it does not merely survive that pressure, it lives there, comfortably, its whole life. So the question sharpens, how? How can something so delicate endure a weight that we are told would crush a ship? To begin to answer that, we have to understand what the pressure down here actually is and what it actually does, because almost everything you have been told about it points in slightly the wrong direction. At the bottom of the Tonga Trench, the water presses down with a force of roughly 1,100 atmospheres. An atmosphere is the weight of all the air above you at sea level, the ordinary pressure you live in every moment of your life without noticing it. Down in the horizon deep, that pressure is multiplied more than 1,000 times over. Imagine the weight of something on the order of a tonne, the weight of a small family car, pressing down on every single square centimetre.
Not on the whole animal, on every square centimetre of it, a patch of skin the size of your fingernail bearing the weight of a car, and the animal beside it and over it and beneath it, all of it pressed from every direction at once by that same colossal weight. It is, by any ordinary reckoning, an impossible place to be alive.
And yet the snailfish swims through it untroubled. To understand why, we have to think carefully about what pressure actually does to a body. And here is where the old story misleads us. We picture pressure as something that crushes, that squeezes, that flattens, like a hand closing around an egg. And that is true for some things, but it is only true for certain kinds of things. It is true for anything that contains a pocket of gas, a pocket of air. Air is springy. Air compresses. Squeeze a balloon and it shrinks. Squeeze it hard enough and it collapses. So any creature carrying air inside it, in lungs, in a gas-filled chamber, is terribly vulnerable in the deep because that pocket of air will be crushed down to almost nothing, and the body around it will be wrecked in the crushing. But water is different. Water does not compress, or very nearly does not. You can press on water with the weight of 1,000 cars and it will barely shrink at all. It simply passes the pressure along evenly in every direction. And the snailfish, well, here is the secret, and the snailfish is made almost entirely of water. Its soft, gelatinous, jelly-like body is mostly water by weight, and water is essentially incompressible. So when the trench presses in on the snailfish from every side with the weight of the ocean, the pressure does not crush it because the inside of the animal pushes back with exactly the same force. The pressure outside equals the pressure inside, balanced perfectly, and the soft body simply holds its shape, untroubled, in equilibrium with the deep. It is not resisting the pressure. It is not fighting it. It is matching it. It has, in a sense, given up the argument and made peace. This is why most fish carry a small gas-filled organ called a swim bladder, a little internal balloon they use to control their buoyancy, to hover at a chosen depth without sinking or rising, adjusting the gas inside it like ballast. It is a clever device and it serves shallow fish beautifully. But down in the trench, a swim bladder would be a death sentence. That pocket of gas would be crushed and ruined and made worthless by the pressure. And so the Hadal snailfish has simply done away with it. It has no swim bladder at all. It has removed from its body the single organ most vulnerable to the deep, and instead it floats on the low density of its own watery, gelatinous flesh, drifting and neutral, needing no balloon of air to hold its place. It has redesigned itself over long ages to ignore the very thing that should destroy it. So that is the first half of the answer to how anything lives so deep. The body is built not to resist the pressure, but to be transparent to it. Soft, water-filled, gas-free, pressed equally inside and out, in quiet balance with the weight of the world. The pressure, it turns out, does not crush a body like this at all. But, and this is the thread that will pull the whole story taut, if the pressure does not crush them, then we are right back to our mystery, and it has only grown sharper, because there is still a danger in all that pressure. It is just not the danger we imagined. The threat is not to the shape of the body. The threat is hidden far smaller and far deeper, down at the scale of the molecules the body is built from, down where life does its quiet chemistry. Pressure that great does something subtle and terrible to the tiny machinery inside every living cell. And how the snailfish answers that threat, and where exactly its answer runs out, is the heart of everything we are descending toward tonight. We have proof, by the way, of just how deep these fish can go, because in recent years we have actually filmed them down there in the dark with cameras lowered patiently into the trenches. The deepest a fish has ever been caught on camera was in the year 2022 in a trench far to the north called the Izu-Ogasawara Trench, where a snailfish was recorded at about 8,336 meters below the surface. 8,300 meters. A soft, pale fish drifting calmly through total darkness, more than 8 kilometres beneath the waves, sensing its world by feel and smell, perfectly at home in a place that would end us in a heartbeat. The deepest fish ever seen by human eyes, or rather, by the patient electronic eye of a camera, since no human eye was there to watch. And that number, 8,300 meters, a little over 8 kilometers, I want you to hold on to it gently as you drift, because it is going to matter enormously. Because the horizon deep, remember, goes down to about 10,800 meters. The deepest fish we have ever found lives at around 8,300. There is a gap, a long, dark gap of more than 2,000 meters between the deepest fish and the deepest point of the trench. 2 kilometers of ocean below the lowest a fish has ever gone. And in all our searching, in all our lowered cameras and baited traps, no one has ever found a fish below about 8,400 meters. Not here, not in the Mariana, not anywhere. It is as if there were an invisible floor hung across the trench at that depth, a ceiling for fish, a line drawn in the dark below, which no fish of any kind has ever been found to live. Above the line, snailfish. Below the line, in the last and deepest stretch of the horizon deep, not a single fish at all. So the mystery has resolved into something very precise and very strange. It is not that life cannot bear the pressure. The snailfish bears it beautifully. It is that something stops the fish at about 8,400 meters, sharp and clean, as if a rule had been written into the water itself. Something draws that line, and whatever it is, it leaves the deepest 2 kilometers of the trench as a realm without fish, a place that belongs to other things entirely, softer and smaller and stranger, things we have not met yet tonight. What is the line? What is the rule? What is the limit, if not the pressure? The answer is waiting just a little further down in the chemistry of the snailfish's own body, in a single small molecule that gives fish their familiar smell and that holds, hidden inside it, the precise and elegant reason why no fish on Earth can ever reach the bottom of the deepest trenches. We are very close to it now, but let us not hurry toward it. Let us drift here a moment longer in the dark with the soft pale fish and the falling silence, and let the question rest gently in your mind as the depth slips slowly past. The snailfish does not look like something built to survive the end of the world. That is the first thing worth sitting with in the dark at this depth. If you had imagined the creature that lives deeper than almost anything else on Earth, you might have imagined something armored, something angular and defended, something that looks like it has been in the pressure and carries the scars of it. You might have imagined something with thick plates, with reinforced fins, with the general bearing of a thing that has learned to resist. The Hadal snailfish is nothing like that. It is soft, it is pale, the color of old candle wax, or of a blush seen through frosted glass. Pink in some lights, nearly transparent in others, as if it had given up the project of opacity altogether and decided simply to let the darkness show through it. It has no scales. Run your finger along its flank, if such a thing were possible, and you would not feel the overlapping armor of a salmon or a cod. You would feel something closer to the skin of a grape, taut, smooth, slightly loose, with a give to it that no freshwater fish has ever prepared you for. Its skin fits it like clothing that is just slightly too large, as if the animal inside has shrunk a little since the cloth was cut. And the body beneath that skin is not what you might expect either. The skeleton of a Hadal snailfish is reduced, partly cartilaginous rather than fully boned, more suggestion than structure. The kind of internal architecture you find in animals that have decided bones are more trouble than they are worth at this depth. Cartilage is flexible, it bends where bone would crack. The The snailfish's internal frame is the frame of something that has, over many millions of years, made a quiet agreement with the water around it. I will not fight you. I will be more like you than like myself. Its body is largely water by mass, not mostly tissue packed with water the way your own muscles are, but water in a more fundamental sense, water as the primary substance, with proteins and cells and the chemistry of life distributed through a medium that is, itself, nearly indistinguishable from the surrounding sea. Pick a snailfish up, and this has happened in the brief window between its capture and its arrival at the surface, and it will begin to fall apart in your hands almost immediately, not because it is fragile in the way a soap bubble is fragile, but because the pressure and temperature at which it is designed to hold its shape are not the pressure and temperature of the surface world, and without those conditions, the whole arrangement begins to dissolve, softly, like something returning to what it was before it was a fish at all. They are, most of them, around 20 to 30 centimetres long, not large, not the giant leviathan the deep might have led you to expect. The snailfish is modest in its dimensions, and this is part of what makes it so quietly strange, that the deepest fish on earth is not a vast and terrible thing, but something you could hold in both hands, something that weighs less than a good-sized apple, something that drifts through 10,000 meters of darkness in a body that is mostly water, wearing a thin coat of biology. Its eyes are reduced. At these depths, this makes perfect sense, and yet it is still a strange thing to understand. The snailfish has given up the one sense that most fish depend on most, because in the Hadal zone, sight is a luxury no one can afford. Below about 1,000 meters, the sunlight is effectively gone. A few wan photons make it that far, just barely, in the clearest of tropical waters, but they carry no useful information. Not enough to see by, not enough to make having eyes worthwhile. Below 1,000 meters, and certainly below the 3,000 or 4,000 meters of the abyssal zone and deeper still, it is dark in a way that no human experience has quite prepared us for. Not the dark of a room with the curtains drawn, not the dark of a moonless night in the countryside. The dark of the Hadal zone is the absolute absence of light, a darkness that has never, in all of geological time, been interrupted by so much as a glimmer of daylight reaching this far down. And so the snailfish's eyes are small and vestigial, and largely decorative in their uselessness. What it has instead are two other senses, both of them exquisitely sensitive, both of them suited to the pressure and the dark. The first is the lateral line, a system of canals and sensory cells running along the flanks of the fish, able to detect the faintest movements and vibrations and pressure changes in the water around it. Not sight, but something adjacent to sight, a feeling of the world rather than a seeing of it, a sense of shapes and movements through the medium itself, the way you might know something was in the room with you by the slight shift of air against your skin. The lateral line tells the snailfish what is moving, what is near, what is disturbing the water in some way that suggests food or threat or simply the presence of another body in the dark. The second is chemoreception, a kind of combined smell and taste, an ability to read chemical signals dissolved in the seawater that surrounds it. In the total darkness of the Hadal zone, chemical signals are everything. A falling piece of organic matter, a dead fish, a fragment of something that once lived at the surface and has been sinking for days, releases chemicals as it drifts down, a slow dissolved plume that extends downward and outward in the current, and a snailfish can follow that plume to its source the way a dog follows a scent trail through a field. And this is how it eats. Not by seeing, not by chasing, but by feeling the faint chemical signature of food in water that has not seen light in 10,000 years. It is, when you allow yourself to imagine it properly, a very quiet way to live. Drifting, sensing, following the faint gradient of something falling from above. moving through the dark, not with the urgent darting speed of a fish in a coral reef, but slowly, with an economy of motion that makes sense when food is rare and energy is precious, and the distance between one meal and the next might be measured in days. Now, the pressure. Because this is where most people's intuition about deep sea life goes wrong, and it is worth correcting gently in the dark, because the truth is so much more interesting than the simple story of crushing force. When you think of pressure at the bottom of the Tonga Trench, you might think of something being compressed, squeezed down, reduced, made smaller and denser by the weight of 10,000 meters of ocean sitting on top of it. The image is of something being crushed. And it is true that the pressure at the horizon deep is extraordinary, roughly 1,100 times the pressure at the surface, or, to put it in a way that might make more immediate sense, about a tonne pressing down on every square centimetre of surface. A small car spread out over an area the size of your thumbnail. It is pressure that would destroy almost any piece of human engineering not specifically designed to withstand it, and it would kill a person almost instantly, not by squeezing them like a tube of toothpaste exactly, but by collapsing any air-filled space in the body, the lungs, the sinuses, the middle ear, and disrupting the chemistry of the tissues in ways that are immediate and catastrophic. Here is the key thing, though, the key thing that the snailfish understood in its slow evolutionary way, long before any biologist did. Water does not compress. Or rather, water compresses so little under pressures far beyond anything that occurs in the natural world that for all practical purposes, including the practical purposes of being a living animal at 10,000 meters depth, water is incompressible. Press on it as hard as you like and it stays the same density, the same volume, the same water. It does not yield and crumple the way air does. It does not collapse the way a gas-filled space does. And this is the snailfish's secret. Its body is water. Not a bag of air with some water in it. Not a system of gas-filled chambers maintaining buoyancy the way a human lung maintains breathing. A body that is so fundamentally water-based, so committed to the liquid medium, that when the ocean presses on it from all sides with 1,100 atmospheres of force, the body simply allows that pressure to be transmitted through it, evenly, without resistance. The pressure outside equals the pressure inside. There is no differential. There is no crushing because there is nothing to crush, no air pocket waiting to collapse, no gas-filled space that the pressure can win against. The snailfish lives at 1,100 atmospheres the way you live at 1 atmosphere, which is to say, it does not notice. The pressure is simply the condition of its existence, the same to it as air pressure is to you, present everywhere, accounted for in every molecule of its body, unremarkable. What this means, and this is the thing worth settling into slowly, is that pressure alone is not what makes the deep dangerous to life. The danger of pressure to a biological system is not the crushing of water-filled tissue. The danger is what pressure does to the chemistry of molecules, and specifically, what it does to proteins. Proteins are the workers of every living cell. They are long folded chains of amino acids that do almost everything that matters in the body: carry oxygen, catalyze reactions, build structure, send signals, digest food. And the thing about proteins is that their function depends almost entirely on their shape. A protein that is correctly folded does its job. A protein that is misfolded or unfolded or distorted in any way does not. It is like a key. The right shape opens the lock and no other shape does, no matter how similar it might look. Pressure at extreme depths does something to proteins. It forces the surrounding water molecules to pack very tightly around the protein surface. Think of the water squeezing in from all sides, filling every small cavity and groove in the protein's folded structure, pressing into the spaces that the protein's shape depends on for its function. This changes the way the protein folds. It pushes the folding toward collapsed, squashed configurations that do not work. It is a very subtle effect at moderate pressures, but by the time you are at Hadal depths, and certainly by the time you are at 8 or 9 or 10,000 meters, the pressure on proteins is significant enough that without some chemical defense, the molecular machinery of the cell simply begins to fail. Enzymes stop catalyzing, structural proteins lose their shape. The chemistry of life, which is almost entirely the chemistry of correctly folded proteins, begins to go wrong in ways that no amount of water-filled body can prevent. This is what the snailfish is actually up against, not the weight of the water pressing on its skin. The weight of the water pressing on the shape of every protein in every cell in its body. And this is where we arrive at last at the molecule. The small, elegant, remarkable molecule that is the actual answer to the actual question. Not how does the snailfish survive the pressure on its body, but how does the snailfish survive the pressure on its chemistry? The molecule is called trimethylamine N-oxide. Biologists abbreviate it to TMAO, which is easier to hold in the mind and easier to say in the dark. And you have almost certainly encountered TMAO before without knowing what it was, because TMAO is the chemical compound responsible for the smell of fish. That particular sharp, salt air, oceanic smell that rises from a market stall or a kitchen where fish is being cooked. That is largely TMAO, or rather it is the breakdown products of TMAO, which the bacteria of decay convert into trimethylamine, the molecule whose smell is so immediate and so distinctive that we have built an entire set of associations around it. The sea, the harbour, the nets, the freshness, or the lack of it. The fish does not smell of the sea by accident. It smells of the sea because it is saturated with a molecule that the sea has, in effect, demanded it carry. A molecule that is the price of living under pressure. Here is what TMAO does at the molecular level, and here is where the analogy that makes it real needs to arrive. Imagine that the proteins in your cells are like very precise, very delicate origami. Each one has been folded over billions of years of evolution into exactly the right shape for exactly the right function. And imagine that pressure is like trying to do origami inside a vise. The harder you press, the more the paper wants to collapse into a flat, useless shape rather than holding its careful folds. TMAO is the thing that counteracts the vise. It works by interacting with the water molecules around the protein, changing the way those water molecules behave, making them, in effect, less willing to press into the protein's surface and disrupt its shape. TMAO shifts the thermodynamic balance of protein folding so that the correctly folded shape is still the most stable shape, even under enormous pressure. It is a chemical counterweight to the force that would otherwise flatten the molecular machinery of the cell. Biologists call it a piezolyte, from the Greek word for pressure, a molecule that specifically counteracts the effects of pressure on biological chemistry. It is, in a word, a pressure stabilizer, and the snailfish is full of it. The deeper the fish, the more TMAO it carries in its tissues. This is one of the cleaner and more striking patterns in deep-sea biology, a nearly linear relationship documented across many species between the depth at which a fish lives and the concentration of TMAO in its body. A fish that lives at 3,000 meters has more TMAO than a fish that lives at 1,000 meters. A fish that lives at 6,000 meters has more than one at 3,000. The deeper the fish, the higher the TMAO, rising steadily as depth increases, as if the fish was slowly filling with the smell of the ocean as a trade for the right to live in its darkest reaches. And this is where the elegant limit lives. This is the rule drawn in the water. This is the reason there is a ceiling for fish at about 8,200 to 8,400 meters, below which no fish has ever been found. It is not that the pressure becomes too strong, It is not that the body gives way, it is that TMAO, the very molecule that allows fish to survive at depth, has a side effect. TMAO is an osmolyte, a molecule that affects the osmotic concentration of the cell's fluids, the relative saltiness and molecular density of the water inside the cell. As TMAO concentration rises with depth, the internal fluids of the fish become more and more concentrated, denser with dissolved molecules. And there is a point, extrapolated from the data on TMAO concentrations across species at various depths, at which the internal fluids of a fish living at that depth would become isosmotic with seawater. Isosmotic means equally concentrated, equally salty, the same osmotic pressure as the ocean surrounding it. This matters enormously because osmoregulation, the process by which fish control the concentration of their own internal fluids, keeping them distinct from the surrounding seawater, is one of the fundamental things fish do to stay alive. It is not a luxury, it is not an optional function. Fish are not the sea. Their cells require a specific internal chemistry, a specific balance of ions and molecules that is different from the chemistry of the water they live in. Maintain that difference, and you are a fish. Lose it, and you are not. If a fish's internal fluids became isosmotic with seawater, if the concentration inside matched the concentration outside, the fish would lose its ability to regulate what moves in and out of its cells. Water and dissolved molecules would flow across the cell membranes according to the laws of osmosis, rather than the needs of the animal. The fish would, in a sense, begin to dissolve into the sea, not literally, not all at once, but in the chemical sense that the careful boundary between inside and outside would be gone, and with it the possibility of being a separate biological system at all. And so the snailfish cannot simply add more TMAO to go deeper. It cannot fill itself up past the level that isosmotically matches the sea. The molecule that protects it is the same molecule that limits it. The cure sets the ceiling. This is what was filmed in the Ogasawara Trench in 2022. A Hadal snailfish at about 8,336 meters below the surface, moving through the dark, which is the deepest any fish has ever been recorded on camera. 8,336 meters. Pale, gelatinous, soft, swimming slowly in the way a snailfish swims, which is to say, almost drifting, a kind of controlled falling through water rather than the propulsive movement of a fish with something to prove. That fish, in that moment, was probably near the edge of what is biochemically possible for any vertebrate on Earth, not because the pressure threatened to break it apart, but because its TMAO concentration was approaching the limit beyond which the chemistry of being a separate organism from the sea itself would begin to fail. Below that depth, below roughly 8,400 meters, the final 2,400 meters or so of the horizon deep are something else. A fishless realm. Not empty, not silent in the sense of lifelessness, but without fish, without vertebrates of any kind, without the things we most easily recognize as animals in the way we are animals ourselves. Whatever lives in those deepest reaches belongs to a different order of existence, and the snailfish, for all its remarkable adaptations, for all the elegance of its water body and its TMAO stabilized proteins and its lateral line sensing and its chemoreception, the snailfish stops here. We are drifting now here in the dark, a little above the invisible line. The snailfish is somewhere nearby, perhaps one of those 20-centimetre pale forms, moving slowly through the water column, reading the chemical signature of something that fell from the surface days ago, following the gradient down toward the trench floor, where food sometimes accumulates. It does not know it is near the edge of anything. It does not know that below it lies a stretch of ocean that no fish will ever reach. It knows only the cold and the pressure that it does not feel, and the faint smell of food in the water, and the slow, continuous, unremarkable fact of being alive in the dark. There is something deeply peaceful about the snailfish when you allow yourself to see it clearly. It is not a creature in agony. It is not struggling against conditions that were never meant for it. It is a creature that has, over time so long, it makes the whole of human history look like a single afternoon, settled so completely into its world that it has become almost indistinguishable from it. Water in water, darkness in darkness. The pressure, its own native pressure, no more hostile to it than the light and warmth of a summer morning, is hostile to you. It lives here as simply and completely as anything lives anywhere. And yet, just below it, just past the place where the chemistry of its own survival draws the line, something is there. Something other than fish. Something that has found a different answer to the same problem, or found that the problem framed differently was never the barrier it seemed. The question is open now, sitting in the water with us unhurried. If not fish, then what? If not the snailfish's gelatinous grace, then what shape does life take in the last 2,000 meters, in the deepest and darkest and most pressured place in the Southern Hemisphere? What fills the gap that chemistry has cut in the column of life, in the space between the last snailfish and the sediment of the horizon deep? We are not ready to answer that yet. The trench goes further. The dark goes further. And somewhere below, past the fish ceiling, the answer is already there, patient and strange, waiting in the way that things in the Hadal zone wait, slowly, without urgency, in the cold and the pressure and the silence that has never, not once in all the deep history of this place, been broken by a sound that did not belong to the sea itself. Let the question drift with you a little longer. Let the snailfish have its last moment in this part of the dark, pale, gentle, astonishing, before the depth takes us past the place where it can follow. The water is cold here. The pressure is exact and constant. The silence is the kind that settles rather than unsettles, the kind that feels less like absence and more like a very long and patient presence. And we are going deeper still. And so we pass the threshold, not with any ceremony. The trench does not mark the boundary. There is no line the water draws, no shift in color or current, no sudden alteration in the quality of the dark. The dark here has been absolute for thousands of meters already. What changes below roughly 8,400 meters is simply this: the fish are gone. And what remains is something older, stranger, and in its own way more astonishing than anything with a spine. The first thing to understand is the body of the Hadal snailfish, because it is worth dwelling on before we leave it behind entirely. Worth returning to it one more time in the way you return to something before a long journey, taking a last clear look. It belongs to the family Liparidae, the snailfishes. A wide and varied family, most of whose members live in shallow coastal waters, unremarkable, easily overlooked. But the Hadal members of this family have been changed by their depth in ways that are visible to the eye, immediately, even in the dim and grainy footage of a lander camera descending on a cable through 6 miles of water. They are pale, nearly translucent in some specimens. The skin has a loose, gelatinous quality, slightly baggy, as though the animal does not quite fill its own outer layer. And in a sense it does not, because the density of its body, the water-heavy tissue, the reduced and partly cartilaginous skeleton, these are not the body of a fish evolved toward firmness and speed. They are the body of a fish evolved toward a single, overriding demand, which is to not die at a depth that kills almost everything else. They have no swim bladder. This is perhaps the most important absence in the animal, the emptiness that defines it. A swim bladder is a gas-filled organ, and gas is the one thing that pressure cannot forgive. At 1,100 atmospheres, roughly a tonne pressing on every square centimetre, more than 1,000 times the weight of the atmosphere you are breathing now, a gas-filled space collapses. There is no negotiating with it. The physics is absolute. The snailfish solve this problem not by strengthening the container, but by removing it entirely, replacing the gas with tissue that is mostly water, and water does not compress. The pressure inside the fish's body equals the pressure outside. The fish is not crushed because there is no differential, no inside pushing against outside, no trapped gas being squeezed toward nothing. The water-filled body sits in the water the way a cup of sea sits in the sea, at equilibrium, at rest, under no particular mechanical strain. The danger, then, is not structural. The danger is chemical. And this is the subtlety that the trench teaches slowly, the lesson that takes the full depth of the water to learn. Pressure does something to molecules. It crowds them. At 1,100 atmospheres, the water molecules surrounding a protein are pressed closer together than they would be at the surface. And this closeness changes how those molecules arrange themselves around the protein's surface, which changes the forces holding the protein in its correct shape, which, if you let it go far enough, begins to distort the protein, to unfold it, to render it something other than what it was meant to be. An enzyme that folds one way at sea level begins, under sufficient pressure, to fold a different way. And when an enzyme folds wrong, it does not work. When enough enzymes stop working, the chemistry of life stops working, and the animal dies, not crushed, not imploded, simply chemically undone. The snailfish carries an answer to this inside every cell of its body, dissolved into its tissues at a concentration that rises steadily the deeper the animal lives. A small molecule, trimethylamine N-oxide, known to biochemists as TMAO. You may have encountered this compound before without knowing its name, because it is the compound that gives fish its characteristic smell, that particular salt and ocean scent that follows a piece of fish left too long in warm air, the smell of the sea brought inland. In shallow fish, TMAO is present in small quantities, a byproduct of metabolism, largely incidental. In Hadal snailfish, it is present in quantities that are anything but incidental. It is structural. It is load-bearing. It is, in the most precise sense, why the fish is alive. TMAO is what biochemists call a piezolyte, a pressure-counteracting osmolyte, a molecule that stabilises proteins against the crowding force of high pressure that interposes itself between the crushing weight of the water column and the delicate architecture of folding enzymes, and holds that architecture together. The concentration of TMAO in a fish's tissue rises in a near-linear relationship with the depth at which that fish lives. Surface fish, low TMAO, fish at 2,000 meters, more TMAO, fish at 5,000 meters, more still. Hadal snailfish, the deepest fish ever placed on camera, filmed at approximately 8,336 meters in the Tonga Trench in 2022, in footage that remains the deepest confirmed fish observation in the scientific record, carry TMAO concentrations high enough to keep their proteins correctly folded and their cellular machinery running at a depth that would dissolve the chemistry of any unprotected vertebrate within minutes. And yet the mathematics of the molecule draws its own conclusion. TMAO is not only a pressure stabilizer, it is also osmotically active, which means it contributes to the saltiness in the osmotic concentration of the fish's internal fluids. A fish needs to regulate the balance between the saltiness of its body and the saltiness of the surrounding seawater. This is osmoregulation, and it is one of the fundamental ongoing tasks of being a fish. Add enough TMAO to stabilize proteins under 8,000 meters of pressure, and the internal fluids of that fish begin to approach the osmotic concentration of seawater itself. Approach what is called the isosmotic point, the moment at which the fish's insides are as salty as the ocean outside. Past that point, the fish cannot regulate itself. The osmotic gradient that allows it to control what enters and leaves its cells reverses or disappears, and the fish begins, in effect, to merge with the sea around it. The calculation, extrapolated from the observed depth versus TMAO relationship across species, places this isosmotic ceiling at roughly 8,200 to 8,400 meters, and the deepest fish ever found hovers just at the top of that range. It is one of those facts that sits quietly in the mind and slowly expands. The snailfish has pushed as deep as the chemistry of its own solution will allow. It cannot go deeper, not because the pressure would crush it. It is not being crushed. Its water-filled body sits in perfect mechanical equilibrium with the surrounding water. But because the very molecule that makes depth possible becomes, past a certain concentration, the thing that makes greater depth impossible. The solution is also the limit. The adaptation is also the wall. Below 8,400 meters, then, there are no fish, not because the trench forbids them with brute force, but because the molecular mathematics of survival draw a line, and the line happens to fall there, and the trench continues for another 2,000 meters past the line, down to the horizon deep at approximately 10,800 meters, and in all those 2,000 meters between the last snailfish and the sediment at the bottom of the Southern Hemisphere's deepest point, no vertebrate has ever been found. The question that floated with us from the end of the last movement settles now into something more specific. Not just what is there below the fish, but what kind of life operates without the molecular toolkit that fish are built around. What finds the last 2,000 meters of the horizon deep not a barrier, but simply a habitat, cold and pressured and dark and ancient, no different in principle from any other place where living things have decided over sufficient time to make their arrangements? The answer is already waiting in the sediment, and the sediment is not silent. That is the first thing worth saying about the floor of the Tonga Trench below the fish ceiling, below the last place any vertebrate has been reliably found, in the 2,000 meters of cold, dark water that sit between the TMAO limit and the actual bottom of the horizon deep. It is not a void, it is not a lifeless mineral plane where nothing moves and nothing grows, and the pressure has simply won. The sediment is active, it breathes in its own chemical sense, it receives, it processes, and the animals that live inside it and on its surface are, if anything, stranger and more interesting than the snailfish they will never meet, because they have been shaped by an even more extreme version of the same pressures and have arrived at solutions that a vertebrate body plan cannot even approximate. Begin with what falls. Everything that lives at Hadal depth, everything that is not a microbe synthesizing energy from chemical reactions in the sediment itself, depends, ultimately, on the surface world, on the sunlit zone far above, where phytoplankton bloom in warm water and copepods graze on the blooms and fish eat the copepods and whales eat the fish, and the whole bright, productive machinery of the upper ocean runs on sunlight and dissolved gases. None of that reaches 10,000 meters down. The light is gone by 1,000 meters. The warmth is gone not long after. What arrives at the trench floor is not light and warmth, but the consequence of light and warmth, the slow, continuous, immeasurably dilute rain of organic matter that oceanographers call marine snow. Marine snow is exactly what the name suggests, if you allow that snow can fall in complete darkness through water that is close to freezing, and that the flakes are not ice crystals, but the crumbled remains of life. Dead phytoplankton cells, their cell walls beginning to dissolve. fecal pellets from zooplankton, small enough to be nearly invisible, dense enough to sink rather than float. Fragments of mucus secreted by gelatinous animals higher up, now broken loose and drifting. Shed exoskeleton fragments from the vast populations of crustaceans in the midwater. The stiffened bodies of single-celled organisms whose mineral tests, their tiny shells, are heavy enough to carry them downward through the water column, even as the organic matter inside them is being consumed by bacteria. All of this falls slowly. A particle of marine snow may take weeks to travel from the sunlit surface to the trench floor, and during those weeks it passes through layer after layer of hungry things: bacteria, deep sea fish, midwater invertebrates, each of which consumes a fraction of the organic carbon and releases the rest, slightly diminished, to continue downward. By the time a particle of marine snow reaches the abyssal plain at 4,000 or 5,000 meters, the vast majority of its organic carbon is gone, consumed on the way. By the time it falls another 5,000 or 6,000 meters to the Hadal zone, what arrives is a ghost of the original, a trace of carbon in a mineral husk, not rich food by any measure, but food nonetheless. And in a place as food-poor as the horizon deep's floor, even a trace is enough to sustain a community built around patience. This is why the deepest point is not the most crowded. It is simply a consequence of arithmetic. The deeper you go, the less food arrives, because the longer the journey, the more is eaten en route. The floor of the Tonga Trench at 10,800 meters is one of the most food-starved habitats on the surface of this planet, receiving perhaps a few milligrams of organic carbon per square meter per year, a quantity so small that if you tried to weigh it in your hand you would feel nothing. And yet the entire economy of the deepest community rests on it. And yet there is a complication. And the complication is the shape of the trench itself. A trench is not flat. A trench is a V, or something close to a V, steep walls converging toward a narrow axis at the bottom, walls that in the Tonga Trench reach angles steep enough to be genuinely dramatic, walls down which sediment does not sit quietly, but slides, tumbles, slumps and falls. And as sediment slides down the walls of a V, it is not evenly distributed across the bottom. It converges toward the axis. It concentrates. The trench shape does something that seems simple, but has a significant consequence. It takes the organic material that settles across a wide area of the surrounding abyssal plain, material that would, on a flat seafloor, be spread thin across thousands of square kilometers, and it funnels it toward the narrow bottom of the trench, the same volume of falling material concentrated into a much smaller area. The result is that certain stretches of trench floor at the very axis of the V can be locally richer in organic carbon than the surrounding abyssal plain several kilometers shallower. The funnel collects, the bottom of the V accumulates, not everywhere, not uniformly. The trench is long and the funneling is irregular, interrupted by the uneven topography of the walls. But in principle and often in practice, the axis of the Tonga Trench can concentrate food rather than starving it away. The trench floor is, in this sense, a trap, and the trap catches not only sediment but the carbon encoded in it. There is another source of food delivery to the trench floor that operates on a different timescale entirely, and it is intimately connected to the fact that you are standing, in a sense, above one of the most seismically violent places on Earth. The Tonga Trench exists because the Pacific Plate is sliding beneath the Tonga Plate at roughly 24 centimeters per year at its northern end, a convergence rate so fast that it is among the highest measured anywhere on the planet, fast enough that geologists studying plate tectonics use the Tonga system as something close to an extreme example of what subduction can become when the forces involved are sufficiently large. 24 centimeters per year is, in the context of geology, not slow. it is fast enough to generate frequent and powerful earthquakes, and those earthquakes do something to the sediment on the trench walls. They shake it loose. When the seafloor moves, when the Pacific plate lurches another fraction of a centimetre downward, and the overlying water column shudders and the walls of the trench vibrate, the sediment that has been slowly accumulating on the slopes is disturbed. Some of it slides, some of it flows, becoming a turbidity current, a dense slurry of sediment-laden water that rushes down the wall like an underwater avalanche, gathering speed and mass as it descends, until it reaches the trench floor and spreads across the axis, depositing a layer of fresh sediment, rich in relatively undecomposed organic material that has not had time to be consumed on the slow drift down from the surface. These earthquake-triggered sediment slides are pulses of food. They arrive suddenly, irregularly, in quantities that can be large relative to the baseline trickle of marine snow, and they represent a mechanism by which the tectonic violence of the subduction zone, the same violence that created the trench, the same violence that is, over geological time, slowly consuming the Pacific plate, periodically refreshes the food supply at the trench floor. The same force that built the habitat also feeds it in bursts between the long, patient silences of the slow carbon rain. The community that lives on and in the sediment at the axis of the horizon deep has evolved to take advantage of both, the slow background rain and the occasional pulse, patience and opportunism held in the same body. The most conspicuous opportunists are the ones that arrive in swarms. Hirondellea. The genus name carries the sound of something light and darting. In French, hirondelle means swallow, the bird, which is, in its way, accidentally appropriate because Hirondellea amphipods at the trench floor move with a speed and urgency that seems almost out of place in a world where everything else is slow and cold and deliberate. They are crustaceans, distantly related to the sandhoppers you might find under seaweed at the beach, and in their basic body plan they recall those familiar animals, segmented, many-legged, curved into a slight comma shape when at rest. But what Hirondellea has become in the Hadal zone is something considerably stranger than any beach hopper. Hirondellea gigas is the species most closely associated with the deep trenches of the Pacific, and Hirondellea dubia is found in the Tonga system specifically, the twin trenches that together represent the deepest cutting of the seafloor in the Southern Hemisphere. Both are small by the standards of things you encounter in ordinary life. A few centimetres long, the kind of animal you might overlook if you came across it unexpectedly. But at Hadal depths, they are not small at all. They are, in fact, larger than their shallow water relatives, following the pattern that researchers have noted in various deep-sea crustaceans and called deep-sea gigantism. The mechanism behind this gigantism is still debated, but the observation itself is consistent. In the cold, food-limited, high-pressure waters of the deep, some invertebrate lineages grow larger than their shallow counterparts, perhaps because cold temperatures slow metabolic rate and allow longer growth periods, or because large bodies are more efficient at fasting between infrequent food pulses, or both. When food arrives at the trench floor, when a fish carcass falls through 10,000 meters of water and settles into the sediment, or when a seal or whale descends through its own death and comes to rest at the axis of the V, the Hirondellea do not wait patiently. They arrive in numbers that are difficult to imagine if you have not watched the footage from baited camera traps lowered to Hadal depths. Hundreds of them, sometimes thousands, converging from across the sediment in the darkness, drawn by chemoreception, by the dissolved chemical signal of decomposing organic matter spreading outward through the still water of the trench floor, arriving within minutes to hours and covering the carcass so completely that what had been a fish becomes, in the footage, a writhing mass of small, pale bodies working in what looks almost like coordinated urgency, but is simply hunger meeting opportunity. A fish that took years to grow, that swam thousands of kilometers in its lifetime, that knew the photic zone and the mesopelagic zone and perhaps the upper reaches of the abyssal, that fish arrived dead at the trench floor, is stripped to bone in a period that researchers measuring with baited cameras have described as startlingly fast. The scavengers of the horizon deep are efficient in the way that very hungry things in very food-poor places tend to be efficient. Nothing edible is left. The bone itself, rich in lipids, will be processed by specialized bacterial mats that can dissolve it slowly over longer periods. But the soft tissue, the muscle and fat and organ, is claimed almost immediately by Hirondellea, and the event that might have sustained the community at a slow, distributed rate for weeks is instead concentrated into a brief collective feast. This is one of the ways the trench floor feeds itself. Feast and fast. The pulse of an earthquake-loosened sediment slide, or the windfall of a falling carcass, or the trickle of marine snow between events, and in every interval, patience. But what Hirondellea can do that makes it genuinely unusual, even by the standard of unusual Hadal adaptations, is not the swarming. It is what it does when the carcasses are absent and the marine snow is too thin to sustain a community of hungry crustaceans. What it does when the only material arriving at the trench floor in any quantity is material that nothing else at depth can digest. Hirondellea gigas can eat wood, not metaphorically, not partially, not with assistance from gut bacteria, in the way that many animals use symbiotic microorganisms to process difficult substrates. Hirondellea gigas produces in its own gut enzymes capable of breaking down cellulose, the structural carbohydrate that forms the walls of plant cells and makes wood fibrous and rigid. Cellulose is one of the more chemically stable biological materials on Earth. It is what makes wood hard and persistent. Most animals cannot digest it at all, which is why herbivores that eat leaves and grass rely on specialized gut chambers and microbial communities to break it down for them. But Hirondellea gigas has internalized at least some of this chemistry, producing celluloses that allow it to extract energy directly from the cellulose in sunken wood. Think about what that means in the context of where this animal lives, 10,000 meters below the surface of the Pacific Ocean, kilometers below any tree, any forest, any plant capable of producing wood, and yet wood reaches the trench floor, logs and branches that fell into rivers and were carried to the sea. Driftwood that spent years floating on the surface until it finally waterlogged and sank. The structural remains of seagrasses, of kelp holdfasts, of every kind of plant material that the surface ocean and its margins shed constantly into the water column. All of this sinks eventually, and some fraction of it, waterlogged, softened, dense enough to continue falling through the entire water column, arrives at Hadal depth, and Hirondellea gigas is waiting for it with the biochemical tools to do what almost nothing else in the deep ocean can. It is one of those adaptations that stops you if you dwell on it. An animal built for the deepest, darkest, most pressure-crushed environment on Earth, carrying in its gut the means to digest the products of forests it will never be near, exploiting a food source that drifts down through 10,000 meters of ocean because the surface world and the Hadal world are connected by gravity and time, and the connection, however thin and slow, is enough. There is another thing that some Hadal amphipods carry, a different kind of material incorporated into their bodies, this one not eaten for nutrition, but built into the structure of the exoskeleton itself. Researchers examining Hirondellea in the deep trenches have found evidence that these amphipods can sequester aluminium from the sediment and incorporate it into a kind of protective coating or gel on the outer surface of their shells. Aluminium, the the same light metal that makes up a significant fraction of the Earth's crust, abundant in many mineral forms, present in the sediment of the trench floor as dissolved ions and mineral particles. The amphipod takes it up and builds with it. The purpose, or at least the proposed purpose, involves the chemistry of the deep water itself. At great depth, the water becomes increasingly corrosive to calcium carbonate, the mineral that most marine organisms use to build shells, bones, and other hard structures. The pressure and the temperature and the chemistry of very deep water collectively drop the saturation state of calcium carbonate below the point where it is stable, meaning that calcium carbonate structures begin to dissolve rather than persist. For animals that have built their protective exoskeletons from calcium carbonate, the deep ocean represents not just a pressure challenge, but a corrosion challenge. Their armor dissolves. Hirondellea's exoskeleton is not made of calcium carbonate. It is made of chitin, which is not susceptible to this dissolution. But the aluminium coating may provide an additional layer of chemical protection, a mineral armor built from the materials the trench itself provides, adapted to the particular corrosive chemistry of the Hadal environment. The trench floor becomes, in this reading, not just a habitat but a construction material, the animal drawing what it needs from the sediment that surrounds it. And then there are the Xenophyophores. If the snailfish is the vertebrate representative of the deep, gentle and gelatinous and pale, and the Hirondellea amphipod is the crustacean representative, energetic and numerous and chemically resourceful, then the Xenophyophore is the representative of something older and simpler and, in certain respects, more astonishing than either of them. It is a single cell, one continuous membrane enclosing one protoplasm, one living unit in the same basic sense that a bacterium or an amoeba is a single living unit, except that a Xenophyophore can grow to 10 or 20 centimetres across, the size of your open hand and sometimes larger. A single cell you could hold, if you were careful, if the pressure and temperature and chemistry of 10,000 meters of ocean was somehow absent, which of course they are not. The Xenophyophores are foraminifera, a group of mostly single-celled organisms with a deep evolutionary history, some of whose smaller relatives build beautiful mineral shells and drift through the upper ocean in numbers vast enough to form significant geological deposits when they accumulate on the seafloor over millions of years. The foraminifera as a group are ancient and diverse and ecologically significant, but most of them are microscopic. The Xenophyophores are the exception. They are the foraminifera that decided over evolutionary time to become enormous. A Xenophyophore does not have a skeleton in the way that an animal has a skeleton. It does not have bones or cartilage or any internal mineral structure. Instead, it builds what is called a test, which is a fragile external structure that serves something like the function of a shell, providing both support and partial protection. And the way it builds this test is one of the things about Xenophyophores that repays attention, because it builds it from whatever is available. It takes sediment grains from the trench floor and glues them together. It takes the mineral shells of foraminifera that have fallen from the surface and glues those together too. It takes fragments of other organisms' hard parts, spicules from sponges, debris from the surrounding matrix, and adds them to the structure, building an agglutinated architecture that is literally assembled from pieces of the trench itself. The organism constructs its home from the material that the trench has collected, from the slow accumulation of the surface world's cast-off minerals settling over geological time. The test is fragile. If you could pick one up, if you could somehow bring one to the surface intact, which is difficult because the structures are delicate and because the transition from 10,000 meters of pressure to surface pressure disrupts biological material in various ways. It would be a loose, irregular, slightly granular thing, not the tight spiral of a snail shell or the symmetrical geometry of a sea urchin test, but something more improvised looking, assembled rather than grown, which is in fact exactly what it is. assembled, piece by piece, grain by grain, from the floor of the horizon deep. Xenophyophores have been recorded among the deepest habitats in the trenches, part of the community that persists in the sediment-covered axis of the V, where the fish cannot go and where even the amphipods must wait between feasts. They are, in their way, patient organisms, single cells operating on the deep ocean timescale, which is not the timescale of the snailfish hunting a prey item or the amphipod rushing to a carcass, but something slower, more geological, more in keeping with the pace of the marine snow itself. And they are ecosystem engineers. This is a term ecologists use for organisms that, through their own physical presence and activities, modify the habitat in ways that create conditions for other species. Beavers building dams, elephants knocking over trees, reef-building corals constructing entire geological structures from their own skeletons. Xenophyophores are ecosystem engineers at the scale of the individual cell. Their tests, their agglutinated shells, create three-dimensional structure in a habitat that would otherwise be largely flat sediment. They create crevices and surfaces and microhabitats within their irregular structures, and in those microhabitats live smaller organisms that use the Xenophyophores test as shelter. In a place where structure is scarce and the sediment stretches flat in every direction, and there is nothing to hide behind and nothing to hold on to except the sediment grains themselves, a Xenophyophores test is architecture, it is shelter. Other organisms live inside it and around it, and the single cell that built it becomes, by its mere existence, a community center. A single cell the size of your hand, built from pieces of the trench floor, sheltering communities of smaller organisms in the darkness of the horizon deep. There are moments in the biology of extreme environments when the mechanisms involved are strange enough that stating them plainly is sufficient, and no dramatization is needed or appropriate. The Xenophyophore is one of those moments, and the Tonga Trench does not stop being strange when you lift your attention from the individual organisms and look at what the whole system is doing. Consider what a trench is at the largest scale. It is a place where two tectonic plates meet, and one descends beneath the other, and the descending plate carries with it whatever has accumulated on its surface during its long journey across the ocean floor: sediment, mineral crusts, and organic carbon. The carbon that arrived at the seafloor as marine snow that was processed by microbes and incorporated into sediment, that accumulated in layers over millions of years, and that now finds itself at the trench axis, not simply resting on the seafloor, but being actively carried downward into the earth by the motion of the subducting plate. The Tonga Trench is, among its other identities, a carbon burial machine. Trenches generally, and the Tonga Trench specifically because of its extreme depth and its active high-rate subduction, bury organic carbon at rates that are disproportionate to the tiny area of the seafloor that trenches actually occupy. The total area of all Hadal trenches combined is a small fraction of the ocean floor, but their contribution to carbon burial and to the long-term cycling of carbon through the Earth's surface systems is significant in ways that researchers are still working to fully quantify. At the bottom of the trench, the sediment is rich in organic carbon relative to the surrounding abyssal plain. The funnel effect, the earthquake pulses, the concentration of marine snow at the axis, and that carbon supports a community of microbes whose metabolic activity, whose rate of consuming organic carbon and releasing carbon dioxide is measurably higher at trench bottoms than on the surrounding abyssal floor. The microbes at the floor of the horizon deep are busy by the standards of deep-sea microbial communities because they have more to work with. The food poverty of the deepest point, which limits the animals to scavengers and filter feeders and the occasional cellulose-digesting amphipod, is not poverty for the microbes. The microbes are extracting energy from carbon compounds that the animals have already passed over, from substrates too recalcitrant for a crustacean gut to handle, from the molecular residue of organic matter that has already been processed multiple times on its long descent, and the carbon that the microbes do not consume is buried. It is incorporated into the sediment, compressed under the weight of new sediment arriving above it, and carried downward by the motion of the Pacific plate as it slides beneath the Tonga plate. The trench in this reading is not just a habitat at the bottom of the ocean. It is a slow, patient, geological mechanism for removing carbon from the ocean atmosphere system and returning it to the interior of the Earth. What the surface world sheds, the dead plankton, the fecal matter, the bodies of animals that lived in the sunlit zone and sank when they died, enters the trench, is processed by the community of microbes and invertebrates at the axis, and what remains is carried downward with the plate into the mantle, into the slow geological cycles that operate over tens of millions of years and that eventually return carbon to the surface through volcanic outgassing, completing a loop so large and so slow that it barely registers against the time scale of a human life or a civilization or even the entire Holocene. The Tonga Trench has been doing this for millions of years. It will continue doing it long after the particular arrangement of continents and ocean basins that defines the current geography of the Pacific has been replaced by whatever arrangement comes next. The trench does not notice us. It does not notice the ships that map it with sound, or the submersibles that carry humans down to its floor for a handful of hours, or the baited cameras that descend and ascend and carry back footage of amphipods swarming in the darkness. It processes, it buries, it carries the surface world's carbon into the Earth's interior at 24 centimetres per year, 1 year at a time, in the dark. And there is one more thing the trench carries down with the sediment that is worth sitting with for a moment, because it arrives at the horizon deep from a different direction than the marine snow by a different mechanism, and its presence at 10,000 meters is one of the more sobering facts that the study of the Hadal zone has produced in recent decades. Researchers studying the bodies of Hadal amphipods collected from the Kermadec Trench, the twin system immediately adjacent to the Tonga Trench, the two trenches together forming the deepest cutting of the Southern Hemisphere's seafloor, found elevated concentrations of persistent organic pollutants in the amphipod tissue. PCBs, polychlorinated biphenyls, and related compounds, industrial chemicals that were manufactured and used extensively through much of the 20th century that were found to be toxic and bioaccumulative and that were banned or severely restricted in most countries beginning in the late 1970s. Chemicals that, even after their production largely ceased, remained in the environment because they are extremely stable, resistant to chemical breakdown, and capable of persisting and concentrating in biological tissue over long periods. PCBs in the bodies of amphipods at the bottom of one of the deepest trenches on Earth, at depths where no human being had ever lived or worked, in a place so remote from any industrial activity that the idea of contamination seems almost absurd until you understand the pathway. PCBs entered the ocean through dumping and runoff and atmospheric deposition over decades. They concentrated in marine organisms through bioaccumulation, each step up the food chain concentrating the chemicals further in tissue. Dead organisms carrying PCBs in their tissue sank, became part of the marine snow, fell through the water column and arrived at the trench floor, where the amphipods consumed them. The chemicals do not break down in cold, dark, high-pressure water. They accumulate in amphipod tissue just as they accumulated in the tissue of the organisms that carried them down. The deepest place in the Southern Hemisphere is not beyond the reach of the surface world's consequences. The trench catches everything that falls. It catches the organic carbon of dead plankton and the cellulose of sunken wood and the lipid-rich flesh of fallen fish and the chemical legacy of a century of industrial production. It does not discriminate. The funnel concentrates whatever arrives and, What arrives carries the history of the surface world encoded in its chemistry, not just the natural history of carbon cycling and biological productivity, but the other history, the one written in molecules that were not supposed to be there and that persist because no natural process at Hadal depth knows how to dismantle them. The amphipods swim in the dark and eat what falls and carry in their bodies the traces of a world 10,000 meters above them that they have never experienced and cannot imagine, if imagining is a capacity that belongs to an amphipod. They simply eat, they swarm, they wait, they digest wood, they carry aluminium from the sediment in coatings on their shells. They live at pressures that would reduce any human structure not specifically engineered to withstand them to a crushed fragment, and they do this without a TMAO ceiling to stop them, without a swim bladder to compress, without the protein folding constraints that set the limit on fish because their body chemistry has solved the pressure problem in different ways, or because their body chemistry never evolved the particular vulnerabilities that make fish stop at 8,400 meters. Below the fish ceiling, in the last 2,000 meters of the Tonga Trench, in the cold, slow darkness of the horizon deep at 10,800 meters, the community that exists is built from patience and opportunism and the chemistry of pressure and the slow falling food rain from a world of light that is, in every physical sense, unimaginably far above. Snailfish cannot come here. The molecular mathematics of TMAO run out before the trench does, but Hirondellea is here, and the Xenophyophores are here, their hand-sized single cells assembling shells from the sediment of the trench floor and sheltering smaller lives within them. And the microbes are here, busy in their chemical way with the carbon that has arrived from the surface and will not arrive again for a long time. And the sediment is here, accumulating, being carried downward, carrying with it the compressed organic record of everything the surface ocean has shed into the deep since the trench opened its V into the Pacific seafloor and the Pacific plate began its long, slow descent. And so we rest for a moment at the edge of that boundary. The snailfish, with their pale, translucent bodies and their careful molecular mathematics, move through the water at around 8,000 meters, and below them the trench continues, indifferent to the limit they have reached. The darkness is total, the cold is steady, and the world below the fish ceiling is not empty. It is simply different, built from different architectures, governed by different rules, populated by creatures that solve the problem of pressure and food scarcity in ways that have nothing to do with TMAO and nothing to do with a swim bladder and nothing to do with the particular vertebrate body plan that gave us, eventually, the snailfish. The Hirondellea amphipods are among the most abundant visible animals in the Hadal zone, and they are everywhere in the Tonga Trench in a way that the word everywhere only begins to suggest. Think of them as small, armored, extraordinarily efficient, closer in shape to a shrimp than to anything you might keep in a garden pond, though the garden pond is a useful reference point only for their approximate size and their general crustacean profile, not for anything about their life or their habits or their extraordinary patience. They belong to the order Amphipoda, and the genus that dominates the deepest trenches, Hirondellea, has been found in the Tonga Trench and the neighbouring Kermadec system in the form of the species Hirondellea dubia, and in the broader Pacific Hadal world in the form of Hirondellea gigas, the giant of its kind and the one that has been most closely studied. They are not large by human standards, but they are large by the standards of their order, and that is itself a fact worth dwelling on. Deep-sea gigantism is the phenomenon in which certain invertebrate animals in cold, deep water grow notably larger than their close relatives in warmer, shallower places. The mechanism is debated. Cold water slows metabolism, which may allow animals to live longer and therefore grow larger. The absence of certain predators may remove a pressure towards smallness. The specific chemistry and food availability of the deep may play a role. The exact reasons are not settled, and different species in different environments probably reach unusual size through different combinations of factors. But the pattern is real and documented, and the Hirondellea of the Hadal trenches are among its clearest examples: larger, more robustly built, more imposing than you would expect from their shallow water cousins. When researchers drop bait to the floor of the Tonga Trench, a parcel of fish or a dead animal sent down on a lander, a vehicle designed to sink under its own weight and then, at a signal, release its ballast and float back up with its cameras and its data, the amphipods arrive quickly. The word quickly is doing considerable work in that sentence. They arrive in hundreds. They arrive in thousands. A fish carcass lowered to the trench floor and left for hours can be stripped to bare bone by Hirondellea in a period of time that seems, when you think about the scale of the place and the darkness and the cold and the pressure, almost impossible. The bone remains clean and pale and ghostly on the sediment, and the amphipods have dispersed again into the darkness to wait for the next parcel of food to fall. This is the nature of life at the bottom of the Tonga Trench, and it is worth sitting with it for a moment. The amphipods do not hunt exactly in the way a predator hunts. They are scavengers and opportunists, exquisitely sensitive to chemical traces in the water. The faint molecular signal of something dead, something rich with the energy of the surface world, falling through the darkness toward the floor. Their chemosensory apparatus, the antennae and sensory hairs that pick up these signals, is finely tuned to exactly this kind of detection across distances that are, relative to their body size, enormous. A Hirondellea detecting the chemical trace of a dead fish several meters or tens of meters away in the total darkness of the horizon deep is performing an act of sensory perception that is, in its own quiet way, extraordinary, navigating by smell and chemistry through a world with no visual reference points at all, following a gradient of molecules dissolved in near-freezing water under more than 1,000 atmospheres of pressure, toward a meal that may be the difference between surviving and not surviving for long enough to reproduce. And yet Hirondellea gigas has a further adaptation that seems, when you first encounter it, almost absurd in its context. Its gut produces enzymes capable of breaking down cellulose. Cellulose is the structural molecule of plant cell walls, the thing that makes wood hard and grass fibrous, and the inside of a stick stiff rather than soft. It is one of the most abundant organic molecules on Earth's surface, and almost no animal can digest it directly. Most animals that eat plant material rely on microbes in their gut to do the cellulose breaking for them. But Hirondellea gigas produces cellulase enzymes in its own digestive system without relying on gut microbes for that specific task, and this means that it can eat and digest wood. Wood. At the bottom of the Tonga Trench, kilometres below any living tree, and the wood is there. Sunken driftwood, branches and logs that have fallen into rivers and been carried to sea and eventually sunk, drifting down through the water column over days or weeks or months until they come to rest on the abyssal plain, or, if their trajectory is right, on the sediment of the trench floor itself. Wood falls into the deep ocean more often than you might imagine, and in the Hadal trenches it is a resource. For an animal that can break down cellulose, a piece of sunken wood is a concentrated package of organic carbon, a store of energy that most other animals in the same environment cannot access at all. Hirondellea gigas has found a niche within the already narrow niche of the Hadal zone, exploiting a food source that its neighbors cannot touch, digesting the structural material of forests that existed, perhaps in river valleys thousands of kilometers away and years or decades ago. There is something in this that rewards attention. The trench is not a place of plenty. The marine snow falls slowly, and much of it is consumed or decomposed long before it reaches 10,000 meters. The carcasses of large animals are rare events, brief bonanzas that attract the amphipods in their thousands, and are consumed completely and then are gone. The trench floor, for most of the time, is a place of waiting, and the ability to eat wood, to unlock the energy stored in the structural material of the terrestrial world, drifting down as a kind of gift from the surface, is an adaptation that speaks to the selective pressure of chronic hunger, the evolutionary advantage of being able to extract nutrition from sources that no competitor can use. There are also reports, carefully documented, of certain Hadal amphipods incorporating aluminium from the sediment into a protective gel or coating on their exoskeleton. Aluminium is present in deep-sea sediments in various mineral forms, and the water at great depth is corrosive to carbonate structures. It dissolves shells and exoskeletons made from calcium carbonate with a slowness that is, over geological time, inexorable. An amphipod that can build a protective mineral coating from the sediment around it using aluminium rather than carbonate chemistry is solving a problem unique to its environment in a way that has no equivalent in shallow water. It is the deep's version of local material adaptation, using what is present in the extreme environment not despite the chemistry of that environment, but because of it, in response to it, as a solution to the specific corrosive conditions that exist at depth. And then there is the pollution. This is the part that pauses the wonder briefly, not to alarm, but because it is true and because it matters, and because it is, in its own way, a piece of information about the world that the Tonga Trench holds and will hold for a long time. Persistent organic pollutants, PCBs, polychlorinated biphenyls, and related compounds, the industrial chemicals that were manufactured in large quantities through much of the 20th century and were eventually banned or severely restricted because of their toxicity and their tendency to accumulate in biological tissues, have been found in the bodies of Hadal amphipods from the deepest trenches in the Pacific, not in trace amounts, in concentrations that are measurable and significant, higher in some cases than in animals living in coastal waters near industrial regions. How did they get there? They got there the same way everything gets to the bottom of the Tonga Trench. They got there by falling. PCBs and similar compounds are lipophilic. They dissolve readily in fats rather than in water, and they accumulate in the fatty tissues of animals, moving up food chains and concentrating as they go, a process called biomagnification. In the surface ocean, these compounds accumulate in plankton, and from plankton into small fish, and from small fish into larger fish, and from larger fish into the bodies of marine mammals and seabirds and the other animals that sit at the top of the ocean food web. When those animals die, they sink. Their bodies, carrying the concentrated chemical legacy of decades of industrial production, fall through the water column as the most extreme version of marine snow, a carcass rich in the energy of the surface world, and, sewn into its fat, the persistent chemistry of the surface world's industrial past. At the bottom of the Tonga Trench, the amphipods eat what falls. They eat the whale falls and the fish carcasses and the marine snow. And in the fat of what they eat, the PCBs are present. The amphipods accumulate them in turn, and when researchers recover the amphipods from lander traps and analyze their tissue, they find the compounds there at concentrations that confirm what the physics of the situation already suggested. That the trench, with its funnel shape and its accumulation of everything that sinks, is also an accumulator of everything that humanity has dissolved into the ocean over the past century. The deepest, most remote, most inaccessible place in the Southern Hemisphere is not outside the reach of what happens at the surface. It has never been outside the reach of what happens at the surface. It receives the surface world's gifts and the surface world's legacies with equal impassivity, and it holds them, and it will hold them for a very long time. This is not a note of despair, it is a note of connection. The trench is not separate from the world above, it is continuous with it, joined to it by the slow fall of everything that has ever lived in the surface ocean, by the marine snow that carries carbon and nitrogen and the molecular debris of billions of planktonic lives downward through the water column, by the whale falls and the wood falls and the PCB-laden carcasses and the sediment slides triggered by earthquakes on a subduction zone that has been active for millions of years. The Tonga Trench is part of the ocean. The ocean is part of the planet. And the planet is, in ways we are still mapping, a single system in which nothing that happens in one place is entirely without consequence somewhere else. The Xenophyophores know this in the only sense in which a single-celled organism can be said to know anything, which is that their existence is a response to the conditions of the place, and those conditions are shaped by everything the surface world sends down. They are the largest single-celled organisms on Earth, individual cells that grow to 10 or 20 centimetres across, visible to the naked eye, enormous by the standards of any cell, which usually measures in micrometers rather than centimetres. A single xenophyophore, if you could hold it in your hand without destroying it, which you could not. They are extraordinarily fragile, their architecture calibrated for the stillness and the pressure of the deep, rather than for the handling of surface world organisms, would span your open palm. One cell. No heart, no brain, no organized tissue. A single vast cell with many nuclei inside distributed through its gelatinous interior, carrying on the business of life at a scale that has no equivalent in the microbial world, and no equivalent in the multicellular world either, because it is neither. It is something in between or something beyond both, something that evolution arrived at by a different path entirely. They belong to the Foraminifera, the same broad group of single-celled organisms whose tiny shells, accumulating over millions of years on the ocean floor, make up significant portions of the sedimentary rock we now quarry for building material on land. The fossil record of Foraminifera is extraordinarily rich, and their shells are among the most important archives of past ocean temperature and chemistry that geologists possess. The Xenophyophores are the giant branch of this group, the branch that took the foraminiferal body plan and expanded it somehow to a size that seems to require an explanation beyond simple growth. They build a structure around themselves that researchers call a test. A shell in the broadest sense, though shell implies a hardness and a smoothness that the Xenophyophores' construction does not have. They build it by gathering and gluing together what is available on the trench floor: mineral particles, sediment grains, the tiny shells of other foraminifera, fragments of whatever hard material has settled around them. This agglutinated architecture, assembled from the trench itself, using the material of the trench as building material, creating a structure that is literally made from the place it inhabits, is both a home and a trap and a scaffold, and it is, in its own quiet engineering, a remarkable thing. The Xenophyophore does not manufacture its own shell material the way a mollusk secretes calcium carbonate. It finds what is around it and combines it into a structure, and the structure is unique to its location in the same way that a dry stone wall is unique to the landscape it was built from. And these structures, fragile and intricate and enormous for what they are, become habitat. Smaller organisms live within the Xenophyophores' test. Tiny crustaceans, nematode worms, other single-celled organisms, the small mobile fauna of the trench floor that needs a structure to shelter in, a surface to attach to, a slightly different microenvironment from the open sediment. The Xenophyophore, without doing anything deliberate, without any behavior in the sense that an animal has behavior, becomes an ecosystem engineer. It changes the environment around it by existing in it. A field of Xenophyophores on the sediment of the Tonga Trench floor is not simply a collection of large, single cells. It is a heterogeneous landscape in miniature, a set of microhabitats that would not exist if the Xenophyophores were not there, supporting a community of smaller organisms that depend on the structure the Xenophyophores have built from the sediment of the floor. This is how the trench works, not as a collection of isolated extremists, each independently solving the problem of the deep, but as a community of interdependencies, each organism creating or modifying the conditions that make it possible for other organisms to be present. The amphipods create a landscape of stripped bones and disturbed sediment. The Xenophyophores create a landscape of agglutinated towers and sheltered niches. The microbes create a landscape of chemical gradients, consuming organic matter and releasing the byproducts that other microbes depend on. And all of this, every interaction, every dependence, every community structure, is fueled ultimately by what falls from above. Marine snow is not a gentle metaphor. It is a precise description of a real phenomenon. In the upper ocean, in the sunlit zone where photosynthesis is possible, planktonic organisms live and reproduce and die in numbers that are difficult to hold in the mind. Their bodies, when they die, begin to sink. Fecal pellets from the animals that ate them sink. The mucus-feeding webs that some organisms spin to catch particles, becoming too heavy with their catch, collapse and sink. Aggregates of particles form as they fall, collecting other particles, growing into larger and heavier flecks, sinking faster. This material, organic, nitrogen-rich, carbon-dense, the compressed remnant of the surface ocean's biological productivity, falls continuously day and night, through all seasons, through all latitudes, in every ocean on Earth. And it is being eaten on the way down. Every meter of descent is a gauntlet. The bacteria in the water consume organic particles. The small animals of the mesopelagic zone, the twilight zone between 200 and 1,000 meters, eat the falling snow. The deeper animals of the bathypelagic zone eat what gets through the mesopelagic. By the time the marine snow reaches the abyssal plain at 4,000 or 5,000 meters, a fraction of what left the surface has survived the descent. By the time it reaches 10,000 meters, the fraction is smaller still. The trench floor is, in terms of the energy available to the organisms living there, one of the most food-impoverished environments on Earth. Not the most. There are others. The mid-ocean gyres, where surface productivity is low, the hypersaline environments where few organisms can exist at all, but among the most because the food must fall such an enormous distance to arrive, and so much of it is consumed before it does. And yet the trench is, paradoxically, not as simple as a uniform desert of food scarcity. The V-shape matters. The steep walls of the trench are not just a geometric feature. They are a funnel. Sediment and organic matter that accumulates on the surrounding abyssal plain and on the walls of the trench itself can slide downward, carried by turbidity currents or by simple gravity acting on unstable slopes, and concentrate at the axis of the trench. The trench floor can be locally richer in organic carbon than the surrounding abyssal plain at the same depth, not because more marine snow falls on it, but because the marine snow and the sediment from a large area, all the slopes above it, converges toward the lowest point. The trench collects what falls near it as well as what falls on it, and the earthquakes help. The Tonga subduction zone is one of the most seismically active places on Earth, and the earthquakes that are generated there, by the grinding descent of the Pacific plate beneath the Tonga plate at roughly 24 centimetres per year, a rate that is extraordinarily fast by the standards of plate tectonics, where a few centimetres per year is typical and 24 is exceptional, regularly trigger sediment slides on the trench walls. These slides carry organic matter that has been accumulating on the slopes downward to the floor in sudden pulses. A community living on the trench floor that is, in its daily existence, adapted to long periods of food scarcity interrupted by occasional carcass falls, is also on a longer timescale, punctuated by these seismic deliveries, sudden inputs of organic material that represent a concentrated gift from the sediment banks above, shaken loose by the planet's own structural violence. The carbon that arrives at the trench floor by whatever path it takes does not simply stay there. Some of it is respired by the microbial community, consumed, metabolized, converted back into carbon dioxide and other compounds, cycling back into the ocean's chemistry. The microbial respiration rates at the bottom of Hadal trenches are, when measured and compared to the surrounding abyssal seafloor, surprisingly high. The funneled organic carbon feeds a microbial community that is, relative to the area of the trench floor, unusually active. The trench is a hotspot of microbial metabolism in an otherwise sparse deep ocean environment, a consequence of the same geometry that makes it a physical trap. And some of the carbon is buried. Sediment accumulates at the trench floor over time, layer by layer, and the organic matter trapped within it is progressively buried under new sediment, removed from contact with the water above it, preserved in the record of the seafloor in the way that a document is preserved in an archive, present, retrievable in principle, but no longer circulating, no longer part of the active exchange between the ocean and the atmosphere. The Tonga Trench buries organic carbon at rates that are disproportionate to its tiny area because it receives organic carbon from a much larger catchment than its own footprint, and because its geology, the steep, funneling walls, the accumulating sediment at the axis, is efficient at trapping and burying what it receives. Hadal trenches are, in total, a small but real component of the global carbon cycle, places where the surface ocean's biological productivity is carried permanently into the deep and eventually, over millions of years, into the Earth's interior. Because the trench floor is not the end of the story, the Pacific plate on which the trench floor rests is moving, descending, carried into the mantle at roughly 24 centimetres per year, where it slides beneath the Tonga plate. The sediment on the trench floor, the organic carbon, the shells of foraminifera, the bodies of Xenophyophores, the exoskeletons of amphipods, the bones that Hirondellea has stripped clean, and the PCBs sewn into the fat of every animal that ate what fell, is carried with the plate slowly over millions of years into the interior of the Earth. The carbon that once existed in phytoplankton in the sunlit surface ocean that was eaten and repackaged and expelled and fell as marine snow through 10,000 meters of darkness, that was partially consumed by amphipods and microbes and stored in the sediment of the horizon deep, will eventually, in geological time, be carried down into the mantle and recycled into the planet's interior, re-emerging eventually through volcanoes as carbon dioxide, beginning the cycle again. This is the longest version of a loop that the Tonga Trench is part of. Not just the loop between the surface ocean and the trench floor, which operates on time scales of years and decades, but the loop between the ocean and the Earth's interior, which operates on timescales of millions of years. The trench is a point of connection between the biosphere and the lithosphere, between the living world at the surface and the geological machinery beneath, a seam where the chemistry of life is carried into the body of the planet and eventually returned. It is when you hold all of this together, the amphipods and the Xenophyophores and the microbial communities, and the falling marine snow and the carbon buried in the sediment and the plate descending toward the mantle. Something rather different from the monster's lair that the phrase 10,000 meters of darkness might initially suggest. It is a system, a patient, dark, slow-breathing system shaped by physics and chemistry and biology and geology, all operating together across timescales that range from the minutes it takes a Hirondellea to detect the scent of a carcass to the millions of years it takes the Pacific plate to carry the organic debris of the surface ocean into the Earth's mantle. The trench is not separate from the world. It is the world at one of its most extreme and least visited and most fundamental expressions. And none of the animals that live there are monstrous. This bears repeating, not because it requires defense, but because it is worth dwelling on as you lie here in the dark, which is a different kind of dark from the one 10,000 meters below the Pacific, but dark nonetheless, quiet nonetheless, far from the noise and light of the surface world in its own small way. The snailfish are pale and soft and translucent, drifting through the water column with a gentleness that belies the pressure around them. The amphipods are quick and purposeful and organized, stripping the gifts that fall from above with an efficiency that is beautiful in its precision. The Xenophyophores are vast and still and fragile, building their sediment shell homes in the deepest quiet, sheltering the smaller lives that gather around them. And the microbes are invisible, as they are, everywhere, doing the fundamental chemistry of life in the dark at a molecular scale, cycling carbon and nitrogen and other elements through the bodies of organisms and back into the water, and eventually back toward the surface, as they have done in one form or another since life first appeared on this planet. The horizon deep is about 10,800 meters below the surface of the Pacific Ocean. The RV Horizon, the research vessel of the Scripps Institution of Oceanography, gave it its name when its surveys in the early 1950s measured what was there, a depth of almost unimaginable distance, reached by sound bouncing off the sediment and returning upward, carrying with it the information of how far down the trench floor lay. Sound, not light. Because light cannot reach here, and the only light that exists at this depth is the light that living organisms make for themselves. The bioluminescence of whatever creatures have found a use for it, flickering in the darkness in colors and patterns that no human eye will ever see directly. The trench is older than the particular moment when the RV Horizon measured it. It has been forming for millions of years as the Pacific plate has descended and the subduction zone has ground and generated earthquakes, and the V has deepened. Long before any human instrument touched it, life was there. Hirondellea was there, or its ancestors were, adapting generation by generation to the conditions of increasing depth and pressure, and darkness and cold and food scarcity as the trench grew deeper. The Xenophyophores were there, building their agglutinated tests from the sediment of the floor and sheltering the small community of organisms that gathered in their structures. The microbes were there, as they are, everywhere, doing what microbes do. And the marine snow was falling, as it always has, since the surface ocean first became productive enough to generate a downward flux of organic matter. The surface world's dead have been falling to the bottom of what would become the Tonga Trench, since before the trench had its current depth, since before it had its current name, since before the species that named it existed. The falling is older than the namers. This is perhaps the most settling thing about the Tonga Trench, and about the horizon deep specifically. The sense that it exists in time as well as in space, that its 10,800 meters of depth are not just a distance, but a history, a record of millions of years of biological and geological process accumulated in the sediment layers at its floor, written in the adapted bodies of the animals that live there, expressed in the carbon that it carries into the Earth's interior on the slow conveyor belt of the descending plate. What lives at the bottom of the Tonga Trench is not a collection of curious extremophiles. It is a community shaped by deep time and deep pressure and deep darkness and the inexhaustible patience of evolution, working with whatever the surface world sends down. If you are still awake, let that settle. The amphipods are there now in the dark of the horizon deep as you lie here, waiting as they wait for the next signal. The molecular trace of something falling, something carrying the energy of the sunlit world down into the deep. The Xenophyophores are there, assembling their sediment shells, sheltering the smaller lives that cluster around them. The microbes are there, working their chemistry on the organic carbon that has arrived and will arrive again. The marine snow is falling above them all, grain by grain, particle by particle, carrying the compressed biological record of the surface ocean downward through the water column at a pace so slow it is barely a movement at all. There is a steadiness to it in the end, not the steadiness of nothing happening, but the steadiness of processes that are very old and very patient, unfolding at scales that are simply different from the scales at which our own lives move. The trench will be here tomorrow. The amphipods will be there. The pressure will be what it is, a tonne on every square centimetre, stable, constant, neither increasing nor relenting. The cold will be what it is. The darkness will be what it is. And somewhere in the water column between 10,000 metres and the surface, the pale, translucent body of a snailfish will be moving, held in place by the mathematics of TMAO and the incompressibility of its watery body at exactly the depth where the chemistry works, no deeper, no shallower, going about the patient and ancient business of being alive in the dark. If you feel moved to share where you are listening from tonight, what part of the world, what room, what hour, I would genuinely like to know. The comments are there, and I read them. There is something that feels right about people from many different places listening to the same deep water at the same quiet hour. There are more stories here of the same slow and careful kind, waiting for whenever you want them. You have done enough for one day. Whatever was unfinished can wait. Whatever is still turning in your mind can be set down gently, the way sediment settles through still water, without force, without rush, finding its own level in its own time. Like the old Stoics, let the day go. Tomorrow can be tomorrow's. Good night. Sleep well.